Hello everyone, and welcome to BioScholar. In today's video, we'll be discussing chromosomal aberrations. The word aberration means a deviation from what is normal, expected, or usual. In biological terms, it refers to any change in the normal structure or number of chromosomes. Each species has a specific number of chromosomes. Most higher organisms are diploid, meaning they carry two sets of chromosomes, one inherited from the father and the other from the mother. Sometimes, during cell division, especially during chromosome segregation, abnormalities can occur. These are known as chromosomal aberrations. Chromosomal aberrations are broadly classified into two types. First, structural aberrations, where the structure of chromosomes is altered. And second, numerical aberrations, where the number of chromosomes deviates from the normal count. In this video, we'll focus specifically on numerical aberrations, what they are, how they occur, and what consequences they may lead to. Variation in the number of sets of chromosomes is referred to as ploidy, and it is quite commonly observed in nature. Changes in chromosome number can involve either the addition or deletion of individual chromosomes, or the entire set of chromosomes. Based on this, two major classes of ploidy variations can be identified. First, aneuploidy, where there is a change in the number of individual chromosomes. This means one or more chromosomes are either missing or present in extra copies, but the overall chromosome set remains incomplete. And second, euploidy, where the variation involves the entire set of chromosomes. In this condition, one or more complete sets of chromosomes are added. Let's begin by discussing aneuploidy in detail. Aneuploidy is a condition in which the chromosome number is not a true multiple of the basic chromosome number or genome. Organisms showing such abnormal chromosome numbers are called aneuploids. Aneuploids can be classified into several groups based on the type of chromosome loss or gain. The first type is monosomic. Monosomy is a condition in which an organism is lacking one chromosome from its diploid complement. Let's take the human genome as an example. A normal human cell has 46 chromosomes, which we can represent as 2n, meaning two complete sets of chromosomes. If one chromosome from this diploid complement is missing, the condition is called monosomy. In symbolic form, this is written as 2n-1. In humans, a well-known example is Turner syndrome, where instead of 46 chromosomes, only 45 chromosomes are present. Specifically, there is only one X chromosome instead of two sex chromosomes. If another chromosome from a different pair is also missing, the organism is said to have double monosomy. This means two different chromosomes are absent from the diploid complement. It is written as 2n-1-1, each -1 indicating the loss of a chromosome from a different set. Double monosomy is rare in humans but more common in plants. For example, in wheat, Triticum aestivum, the normal chromosome number is 42. If two chromosomes are missing from two different pairs, then it is called double monosomy. We can write it as 2n-1-1. Until now, we've been talking about the loss of one chromosome from each pair. But what if both chromosomes of the same pair are missing? This means the entire pair is absent, a different condition known as nullisomy. Nullisomy is the condition in which both chromosomes of the same pair are missing from the diploid complement. In symbolic form, it is written as 2n-2, the -2 showing that an entire pair is absent. This condition is generally lethal in humans and most animals, because the loss of a whole chromosome pair removes too many essential genes. However, in some plants, nullisomy can occur and the organism may still survive, though often with reduced vigor. So, monosomy and nullisomy are conditions where chromosomes are missing, but the next two conditions involve the addition of chromosomes instead of their loss. These are known as trisomy and tetrasomy. Trisomy is the condition in which an extra chromosome is added to the diploid complement. This means the organism contains 2 complete genomes plus 1 additional chromosome. It is written as 2n plus 1. For example, in humans, a normal genome is represented as 2n, 2 complete sets of chromosomes. If an extra chromosome is added to chromosome 21, the condition is called trisomy 21, commonly known as Down syndrome. In this case, there are 3 copies of chromosome 21 instead of the normal 2. If extra chromosomes are added to 2 different chromosome pairs, the condition is called double trisomy. It is written as 2n plus 1 plus 1, each plus 1 showing an additional chromosome in a different pair. Double trisomy is extremely rare in humans, but can occur, for example, when there is an extra chromosome 21 along with an extra sex chromosome. If instead of adding extra chromosomes to 2 different pairs, both extra chromosomes are added to the same pair, the condition is called tetrasomy. This means the homologous pair is doubled, so there are 4 copies of the same chromosome instead of the normal 2. In symbolic form, tetrasomy is written as 2n plus 2. In humans, examples include tetrasomy X, where females have 4 X chromosomes instead of 2, and tetrasomy 18p, where there are 4 copies of the short arm of chromosome 18. These conditions are rare and often associated with developmental delays and distinct physical features. So, these are the 4 main groups of aneuploidy. Now, let's move on to euploidy. Euploidy is the condition in which 1 or more complete sets of chromosomes are present in an organism. Euploids can be monoploids, diploids, or polyploids. Monoploids are organisms that contain only 1 set of chromosomes, represented as in. This means they have just a single copy of each chromosome, instead of the usual 2 in diploid organisms. Monoploidy can occur naturally in certain male insects like bees, wasps, and ants, which develop from unfertilized eggs. In plants, monoploids can be artificially produced in laboratories for breeding and genetic studies. Diploids are organisms with 2 complete sets of chromosomes, represented as 2n, 1 set from each parent. Humans are diploid, with 46 chromosomes arranged in 23 homologous pairs. Polyploids are organisms with more than 2 complete sets of chromosomes. They are common in plants, such as breadwheat, Triticum aestivum, which is hexaploid with 6 sets, totaling 42 chromosomes. This was a brief introduction to numerical aberrations. I hope you found it helpful. If you have any doubts, drop them in the comments section. And don't forget to subscribe to BioScholar for more videos. To learn about the structure of chromosomes in detail, click on this video.